electron transport system. It takes place in the inner membrane of mitochondria. This is inner membrane, this is outer membrane, this is intermembrane space, and this is matrix. So electron transport system takes place in inner membrane of mitochondria. There are 5 complexes. This is complex 1 called as NADH dehydrogenase. This is complex 2 called as FADH dehydrogenase or succinate dehydrogenase. This is complex 3 also called as cytochrome BC1. This is complex 4, that is cytochrome C oxidase, and this is complex 5, that is ATP synthetase. Let's start with the process. Here, complex 1, NADH2 get oxidized to NAD and hydrogen. This hydrogen split up into 2 protons and 2 electrons. These 2 electrons are accepted by FMN. From here, electrons are transferred to iron sulfur protein. In this process, 2 protons are pumped from matrix into intermembrane space. Now 2 electrons from iron sulfur proteins are accepted by ubiquinone. Again in this process, 2 protons are pumped from matrix into intermembrane space. So total 4 protons are pumped. Now this ubiquinone take 2 electrons, but ubiquinone is a hydrogen. It takes 2 protons from matrix, and here ubiquinone get reduced to ubiquinol, that is UQH2. Again ubiquinol get oxidized to ubiquinone, and here 2 hydrogen are released. Now in complex 2, as we have seen in citric acid cycle, succinate get oxidized to fumarate. So here FADH2 get oxidized to FAD plus hydrogen. These 2 hydrogens split up into 2 protons and 2 electrons, and these 2 electrons are accepted by FAD. From FAD they are transferred to iron sulfur protein. Now here iron sulfur protein transfer electrons to ubiquinone. It's a hydrogen carrier. So here ubiquinone take 2 electrons from FES, 2 protons from matrix, and it get reduced to ubiquinol. After reaching over here to complex 3, ubiquinol get oxidized. There is removal of hydrogen. This hydrogen split up into 2 protons and 2 electrons. And here again the cycle goes on, here and here. 2 protons from here and 2 protons from here are pumped into intermembrane space through complex 3. The total 4 protons are pumped through complex 3 into the intermembrane space. Now here out of 2 electron, 1 electron goes to cytochrome B, and from here also 1 electron goes to cytochrome B, and 1 electron remains in the quinol cycle here and here. Now since cytochrome B receives 1 electron from here and 1 electron from here, 2 electrons from cytochrome B goes to iron sulfur protein, and from iron sulfur proteins 2 electron goes to cytochrome C1. Now there is a small protein, that is cytochrome C, which is present on the outer side of inner membrane. This cytochrome C carries these 2 electrons from complex 3 to complex 4. Then these 2 electrons goes to copper A, then to cytochrome A, then to cytochrome A3, then to copper B. From here, 2 electrons combines with oxygen and protons. Remember, oxygen is the final electron acceptor, and here water molecule is formed. In this process, 2 protons are pumped from matrix into intermembrane space. Now since the protons are pumped from matrix into intermembrane space, there are more protons here in intermembrane space and less protons in the matrix. This creates electrochemical proton gradient, that is chemiosmosis. So the protons from higher concentration goes to lower concentration. Here there is F0 F0 pumps protons, F1 take the energy and convert ADP plus IP into ATP. So here ATP is synthesized, that is terminal oxidation. Complex 1 pumps 4 protons, complex 3 pumps 4 protons, and complex 4 pumps 2 protons. So here NADH2 and FADH2 are used for synthesis of ATP